You are holding your phone right now. You are probably sitting somewhere safe, warm, and completely unthreatened. But somewhere deep in the back of your brain, something old is still running. Something that was written into your biology long before cities existed, long before hospitals, long before the word medicine meant anything at all. That thing is fear of snakes. And the reason it is still running, still alive in you, is because of something your ancestors did that science is only now beginning to fully understand. They survived. Not just once. Not just lucky. They survived in ways that should not have been possible, and the answer to how they did it will change the way you think about your own body. A snake bite, even today, can kill a person in minutes. The venom of a coastal taipan disrupts blood clotting so severely that the blood simply stops working. The venom of a black mamba moves from the bite site to the heart in under 20 minutes. King cobra venom can stop a human diaphragm, the muscle you breathe with, before a person even has time to feel afraid. These are not ancient stories. These are documented, modern, medical facts. And yet, for the vast majority of human history, there were no antivenom labs, no syringes, no emergency rooms, no doctors, nothing but the body itself and whatever knowledge the group had accumulated over generations. So how did anyone survive? The first thing you have to understand is that envenomation and a bite are not always the same thing. Researchers have known for decades that snakes perform what herpetologists call a dry bite. A dry bite is a bite in which the snake delivers no venom or only a tiny fraction of its venom load. Studies published in journals like Toxicon estimate that somewhere between 20 and 50% of all venomous snake bites in humans involve little to no venom injection. The snake makes a choice. Venom is metabolically expensive to produce. Fangs take energy and time to regenerate if broken. For a snake, biting a large creature that cannot be swallowed is a defensive act, not a predatory one, and defense does not always require full commitment. Your ancient ancestor, running through grass or reaching into a rock crevice, may have been bitten, felt the pain, screamed, and survived, not because of anything they did, but because the snake held back. But that was not always the case, and the body had other plans. Researchers Brian Fry at the University of Queensland and his team have spent years mapping the genetics of venom resistance across different species, including humans. What they found is striking. Certain proteins in human blood plasma have structural features that partially blunt the effect of specific venom components. These are not adaptations in the dramatic sense. They are not built purely to fight snake venom. They are ancient proteins that happen to interface poorly with certain toxins, reducing their efficiency in the human body compared to, say, a rodent or a small mammal. Your blood is not immune to snake venom, but it is not a perfect target either. Millions of years of coexistence with snakes on the African savanna left traces in the human immune system that researchers are still cataloging today. Then there is the inflammation response. When venom enters tissue, the body does not simply surrender. It floods the area with immune cells. It triggers swelling, painful, visible, dramatic swelling. That is actually the body attempting to dilute and wall off the venom before it reaches the bloodstream. That swelling that ancient people saw after a bite was not just a symptom. It was a battle. The tissue around the bite was being sacrificed to slow the poison down. Ancient healers did not know the mechanism, but they saw the outcome. They saw that people who survived the first few hours had a chance, and they built their practices around those hours. What did ancient humans actually do in those hours? The answer is more sophisticated than most people assume. Anthropologist and physician Daniel Moorman spent decades cataloging indigenous plant-based medical practices across cultures worldwide. What he and other researchers in the field of ethnobotany found is that independently across Africa, South Asia, the Amazon basin, and indigenous North America, human groups arrived at strikingly similar responses to envenomation. They applied heat, they used specific plants, and they kept the person still. The stillness was not ritual. It was not superstition. It was biology. When a person moves after a snake bite, the muscles contract and pump. Blood circulates faster. The heart rate climbs. Venom that had been contained in tissue is pushed into the lymphatic system and from there into the blood. The ancient instruction, "Do not run, do not move, lie down and be still," was a piece of empirical knowledge accumulated over countless observations across countless generations. It was science before science had a name. The heat application followed a similar logic, even if the mechanism was misunderstood. Applying a burning torch or heated stone near the bite site damaged tissue, yes, but it also disrupted the enzymatic activity of certain venom proteins. Many snake venom components are enzymes, biological machines built to cut, dissolve, and disrupt. Enzymes are exquisitely sensitive to temperature. The protein structures that make them functional begin to denature, to fall apart above certain heat thresholds. Your ancient ancestor did not know what a protein was, but they had observed over generations that heat near the wound sometimes slowed the spread. And they remembered the plants were perhaps the most extraordinary piece. Ethnobotanist Paul Allen Cox, working across multiple continents, helped document hundreds of plant species used by traditional communities in response to snakebite. When researchers began testing these plants pharmacologically, they found that a significant number of them contained compounds with genuine biological activity against venom components. Plants in the genus Aristolochia, used in parts of Africa and Asia, contain compounds that interfere with phospholipase A2 enzymes, one of the most destructive classes of molecules found in snake venom. The Aristolochia user did not know the name phospholipase. They knew that when they pressed the mashed root against the wound, fewer people died. That knowledge was real, it was earned, and it worked. Across the Amazon, indigenous healers used bark decoctions from trees in the genus Ampelosiziphus and other species in rituals and treatments around bite events. When Brazilian pharmacologists analyzed these extracts, several showed measurable anti-inflammatory and venom-neutralizing properties in laboratory conditions. The chemical logic of the forest had been decoded, one painful death at a time, over thousands of years. There is another factor that your ancient ancestors had, that modern medicine is only recently beginning to appreciate with full seriousness: immunity from repeated exposure. In the same way that your immune system remembers a virus it has fought before and builds antibodies to blunt the next attack, the human immune system can build partial resistance to snake venom through repeated sublethal exposure. Snake handlers in religious traditions across the American South, who have been bitten dozens of times over decades, show measurably altered immune profiles compared to non-handlers. Researchers at Loma Linda University found elevated levels of specific immunoglobulin responses in long-term snake handlers that were not present in control subjects. These were not fully protective. They did not make the handlers immune, but they shifted the body's response curve, reducing the lethality of bites that would have been far more dangerous to a naive immune system. In your ancestral environment, children growing up near snakes, in communities that had lived alongside specific species for generations, would have been exposed, through near misses, through minor bites from non-lethal species, through environmental contact, in ways that primed the immune system over time. The most experienced adults in a group, the elders who had survived the most, may have carried the most developed partial resistance, and when they were bitten again, their bodies had already seen something like it. This is not a guarantee of survival. It was never a guarantee. Snake venom killed ancient humans in enormous numbers. The snake fear circuitry in your visual cortex, the pathway that makes your eyes snap to a curled shape before your conscious mind even processes what it saw, was written into your neurology by exactly those deaths, by the people who did not survive, by the ones whose descendants were the ones who spotted the snake first, who jumped back in time, who passed on a brain wired for speed over anything else. Lynn Isbell, a primatologist at UC Davis, put forward what is now called the snake detection theory, which argues that the primate visual system itself, the extraordinary color vision, the fine depth perception, the rapid pattern recognition that distinguishes primates from most other mammals, evolved in direct response to the predatory pressure of venomous snakes. You can perceive the world the way you do, in part because your ancient ancestors who could not spot a snake fast enough did not live to pass on their genes. The snake shaped your eye. The snake shaped your fear response. The snake shaped the way communities organized their knowledge, their plants, their rituals, their healers. And today, When you pull your hand back from something coiled in the garden before your brain has consciously registered what it is, that is not anxiety. That is not irrationality. That is the oldest piece of software still running in your body, the piece that was written in the grass, under the orange sky of an ancient afternoon, by someone who saw the snake just in time and lived to show their children where to look. The same brain that makes you flinch is the brain that made your ancestors survive. It has not been updated. It does not need to be. It is still running right now while you hold your phone in what feels like perfect safety, watching for shapes that should not be there.